Good afternoon, students. So, welcome back to the classes, biology classes, and we are going to start with the biomolecules. Okay, so biomolecules, new chapter we are going to start in today's session. Understood? And this comes under Unit 3 chapter. Okay, this chapter comes under Unit 3 in biology textbook. If you open unit 3, okay, chapter number 9 will be biomolecules. Okay, so majority this is important and there is no deletion of anything in this chapter. So we have been complete. In third unit, we have completed two chapters. What are they? One is cell we have completed, another one mitosis meiosis, that is cell cycle and cell division we have completed. Today, in that unit, we have left with the biomolecules. So let us see, try to understand this in molecular level. You should understand what are these biomolecules. You are also coming across this chapter in chemistry. Chemistry also biomolecules is there, but biomolecules role in chemistry is separate, and here biology, related to biology, biomolecules we are going to study. Especially somewhat related, we are going to study some chemical structure, composition, some chemistry is also mixed here, but they are linked to the living life. Okay. So now coming to the definition of biomolecules. So if all the living organisms, okay, all the living organisms, if you take all the living organisms, they consist of chemicals. What it consists of? Living life. Internally, I am telling. Internally, they are made up of some chemicals or molecular. Living organisms are made up of either chemicals or molecules. Molecular. They are made up of either chemicals or molecules. Is called biomolecules. What it is called? Biomolecules. So, any living organism presence of chemicals and molecules is called as biomolecules. It is called, understood? What do you mean by biomolecules? Biomolecules are nothing but what? All the living organisms are made up of present of chemicals and molecules. So this makes the biomolecules. So we are going to study some molecules. Bio means life. Some molecules are present in the life. So they are called biomolecules. In this chapter, we are going to study the life and molecules, what all chemicals are there, what are enzymes are there, their structure, their function, everything we are going to study. Next, after this, the molecular, we have to understand these biomolecules in the molecular level. Molecular level we need to understand. So now this molecular level involving, first it is involving chemical nature, it is involving chemical nature, then composition, then mode of synthesis, mode of synthesis, behavior of biomolecules, behavior of biomolecules. Okay, these are, we have to understand these chemicals, molecules in molecular level. Okay, chemical nature, how this like protein if you take. ಅದು chemical nature ಹೇಗಿದೆ ಅದರೊಳಗಡೆ ಏನೇನು ಪ್ರೆಸೆಂಟ್ ಇದೆ ಅದು ಏನು ಸಿಂಥಸೈಸ್ ಮಾಡುತ್ತೆ ಅದರ ಬಿಹೇವಿಯರ್ ಏನು ಅನ್ನೋದು ತಿಳ್ಕೋಬೇಕು. Okay, so whatever enzymes you can take, protein you can take, they are all chemicals which are chemicals and takshana will come across carbohydrate, starch, protein, enzymes, hormones also. Okay, so whatever you are studying, we have to study chemical nature, molecular level we have to understand. Composition, ಏನೇನು present ಇದೆ ಅದು composition, mode of synthesis, ಏನು synthesize ಮಾಡುತ್ತೆ, ಏನು ರೆಡಿ ಮಾಡುತ್ತೆ ಅದರಿಂದ, ಆ chemical ಏನು ರೆಡಿ ಮಾಡುತ್ತೆ, ಆ enzyme ರೋಲ್ ಏನು, and their function, behavior of biomolecules. Next, now one more term we are going to come across, that is called as cellular pool. So just you should know what is cellular pool. Okay, the word itself, pool is nothing but these are made up of more and more number of cells. Now, if you take the cell are having, now if you take one cell, the cells are made up of 40 elements. These are made up of 40 elements. Each cell, they are made up of 40 elements which will combine and recombine. This will undergo the 40 elements combine. After that again they will recombine. Okay, after combining and recombining, what happens here is they will produce 5,000 types of chemicals. This will causes 5,000 types of chemicals will be formed. Cell have only 40 elements. Okay, 40 elements. Now think these are all elements. This will combine with this will combine with this will combine with this. So in that recombining, what happened? 5,000 different type of chemicals will be produced because of the mixture of 40 elements. Because of the recombining 40 elements. Okay, so these carry out 1,000 type of reaction. This carry out, this 5,000 chemical will carry 3,000 reaction. 3,000 reactions occur in cell. Okay, this is called as cellular pool. What do you mean by cellular pool here? So this cell with the help of enzyme. So to tell very easily, cellular, the cell have 40 elements which will again combine, recombine. After that, they will form 5,000 different chemicals. These chemicals will undergo, with the presence of enzyme, the 3,000 reactions will be taking place inside the cell. To tell very simple, what do you mean by cellular pool? In the collection of biomolecules in the cell. The collection of biomolecules, what is the definition? The collection of biomolecules, the collection of biomolecules in the cell is called cellular pool. So this is the definition. Inside the cell, when reactions are going on, combination of the cells are going on. So hence these are called as crystallocolloidal system. The term, the cellular pool inside the cell we can call it as crystallo-colloidal system. So crystallo-colloidal system means what? The plasma membrane is present. The cell is there. The cell is covered by the plasma membrane, and inside the protoplasm is there. Okay, inside it is protoplasm. The protoplasm is enclosed by the plasma membrane. Inside the protoplasm only all the reactions are taking place. So it is called cellular pool. What is cellular pool? The collection of biomolecules in the cell is called as colloidal cellular pool. So this cellular pool is crystallo-colloidal system. It is nothing but the protoplasm enclosed by the plasma membrane is called crystallo-colloidal system. Crystal is different, colloidal system is different. Now, in introduction, you have to understand two terms which is called micromolecules and macromolecules. We have two types, micromolecules and macromolecules. First one. First one, micro. Biomolecules, we have two types on the basis of size solubility. On the basis of size solubility, two types. Biomolecules are of two types. One is called the micromolecules. So these micromolecules are smaller in size. Here are some characters. Smaller, size, low molecular weight, then next one, so then simple structure, high solubility, micromolecules, high solubility, found in gas, liquid, solid form. They are found in gas, liquid, or in the form of solid form. These are the characters. Now, these can be both in organic also. They may be present in both organic or inorganic. Example. Micromolecules are based on their size, two types, micromolecules, macromolecules. Micromolecules are smaller size, having low molecular weight within 1000 molecular weight. Simple structure, high solubility. Soluble is very fast and high, found in gas, liquid and solid form. They may exist both in organic and inorganic substances. Now, micromolecules example can pass through the cell. So these are the found and can be organic or inorganic and can pass through the cell. So like water, minerals, amino acids. Water, example, water, minerals, amino acids, sugar, phosphate, etc. All these examples are considered to be as a micromolecules. What they are called? They are called as micromolecules. Sugar, even nucleotide, phosphate, like lipids, nucleotides. These are some examples. Next, macromolecules. Second type is macro. If these are smaller, because opposite we can see here, these are opposite and these are larger in size. Okay? Larger size. Low molecular weight, here high molecular weight. High molecular weight. Simple, but here it is complex structure. High, here low solubility. Okay, high and in macromolecules, these are low solubility. So these are formed by polymerization of micromolecules. More number of micromolecules, okay, formed by polymerization of these micromolecules will form. Okay. Means loss of water and all. And now they are only same and cannot pass into the membrane. So only these are present both, right? These are present only in the organic. Here they can pass the cell membrane. Here they cannot pass the cell membrane. Here they are organic and they cannot pass the cell membrane, but in case of macromolecules, they can pass the cell membrane. Pass the cell membrane. Next examples if you take. So these are proteins, nucleic acid, polysaccharides. Proteins, nucleic acid, polysaccharide, especially monosaccharide, disaccharide, okay, polysaccharides. These are, and obviously these are formed by the condensation of lipids, nucleotides, amino acids. Okay, already we have mentioned that macromolecules are formed from the micromolecules only. These micromolecules undergo condensation, undergo polymerization, and after condensation, these proteins are formed, nucleic acids are formed, polysaccharides are formed by the condensation of amino acid nucleotides. Okay, so this is about the in biomolecules. Based on their size, we have divided the molecules into two types. One is called micromolecules, another one is called macromolecules. Here micromolecules size is smaller, here larger, low molecular weight, high molecular weight, simple, they are complex, high solubility, low solubility. These are found in everywhere, they are existing in organic and inorganic. But these are formed by the polymerization, condensation of micromolecules. These are compulsory, they will be organic. They cannot pass the cell membrane, but these can pass the cell membrane. This example includes protein, nucleic acid, polysaccharide, which are formed again by the micromolecules. Okay, next, according to your NCRT, the heading is given how to analyze chemical composition. This is introduction. Next heading, write down how to analyze chemical composition. Heading, how to analyze chemical composition. How to analyze chemical composition. Okay, so we have to analyze. If you take living tissues, okay, one flowchart I will give you so that you can understand this topic. How to analyze chemical composition. Composition means if you take the cell, okay, if you take one cell, okay, some chemicals, molecules are there, okay. You have to tell what all chemical compositions are present. You have to analyze. You have to think. Composition ಅಂತಂದ್ರೆ ಈ ಸೆಲ್ ಎದರಿಂದ ಮಾಡಿರೋದು. So to know this, to analyze, we need one procedure. Okay, without that we can't do now one procedure in order to understand various biomolecules found. Okay, analyze means to analyze the various biomolecules present in the living tissues, living cells. We are completely talking about the life. On the life ಅಲ್ಲಿ ಏನ್ ಮಾಲಿಕ್ಯೂಲ್ಸ್ ಇದೆ ಏನ್ ಕೆಮಿಕಲ್ಸ್ ಇದೆ ಅನ್ನೋದು ಅನಲೈಸ್ ಮಾಡಬೇಕು. So we have that particular procedure to know, to analyze this. So now all the living tissues, grind it first. I'll represent that. Living tissue. We are taking living tissue. From this living tissue, we have to analyze what it is made up of. Trichloroacetate. Now this living tissue is treated with the trichloroacetate. That is, it is written as CCl3 and COOH. What it is? Trichloroacetic acid. Acetic acid. First, what we are doing to analyze what chemicals are present in living tissue. Living tissue is treated with the trichloroacetic acid. Now, after treating it with this, we are getting fragments, that is staining with the, here these are staining with cheese. We are getting filtrate, okay, filtrate and retentate. I'll explain one by one what all these steps. So what we are doing here, living tissue is treated with the trichloroacetic acid, and these are stained with the some cheese, cotton and all. Okay, we have to stain them with the cheesecloth or cotton, and we will obtain two fractions. What we are going to get are, these are the two fractions we are going to get. The first fraction is called filtrate. The second fraction is called retentate. What it is called? Retentate. The filtrate is called as what? Called as acid soluble. The filtrate what we are getting, that filtrate is called acid soluble. Retentate fraction what we are getting, that is called acid insoluble. Okay, it is called acid insoluble. So after this retentate, which is chemical present in both the fractions, are analyzed and identified. The chemicals which we are getting after treated with the trichloroacetic acid, we are getting some filtrate. This filtrate is called as acid soluble by analyzing. This retentate is called, some analysis has done. Correct. So because before that we were not knowing anything about this living tissue. First it is treated with the chemical trichloroacetic acid. Again it is helped with the help of staining of the cheese. Okay, or cotton. Some filtrate we are getting, some fractions we are getting, which is named as, one is called filtrate, another one is called retentate. One is soluble, one is insoluble. Both are acid only. And these are acid soluble having molecular weight within the 800, and here they are nearly 8,000. Okay, they are. This soluble, acid soluble contains chemicals 18 in the 800 Daltons, DA and Daltons. So these acid soluble have chemicals of molecular weight 18 to 800 Daltons. Okay, this is one point of the acid soluble. Acid insoluble chemicals with molecular mass, molecular weight 8,000 Daltons. Okay, and these are called what? Biomicromolecules, and these are called biomacromolecules. Okay, only we have studied micromolecules, macromolecules. What are the micromolecules? These are small, these are large, these are low molecular weight, these are high molecular weight, simple structure, complex structure. Okay, these are soluble, these are insoluble. That all characters already we have displayed there. Understood? So what we have studied here? So to know, to analyze, how you are going to analyze what chemicals it is present, the simple method we are going to collect the tissue treated with trichloroacetic acid. These two are combined and we will get, and this is treated with the staining with the cheese or cotton, and we will get two fractions. One is called filtrate, another fraction is called retinate. One filtrate is called acid soluble, another retinate is called acid insoluble. Acid soluble is having the chemicals having the low molecular weight 18 to 800 Daltons, that's it. In case of this, molecular weight is 8,000 Daltons. So above these, what what we are going to call biomicromolecules, and these are called biomacromolecules. Okay, complex. Next, so biomacromolecules are three types: proteins, nucleic acids, and polysaccharides. We have written what are the macro: protein, nucleic acids, and polysaccharides. You can also write here examples. But here one more exceptionally, we are leaving lipid here because lipids is due to non-polar. Lipids are having polar head and non-polar tail. Okay, head and tail. There they were having half face is facing towards water. Fluid mosaic model. In cell we have studied. Head is facing outside, tail are facing inside. Inside, which is tail, is non-polar. They are not facing towards the water. In that condition, except lipid, all except living lipid, nucleotides, water, minerals, amino acids are all what? Acid soluble. Rest, proteins, nucleic acid, which are formed by this, but biomacromolecules are formed by the biomicromolecules only. Okay. Next. After this, next one, we have primary and secondary metabolites. We have after how, so you got the idea about the analysis, right? So next one, primary and secondary metabolites. Primary and secondary metabolites. Primary and secondary metabolites. So what do you mean by primary? The substance which are required for the primary. First one I am explaining. First one, primary are nothing but, metabolites are nothing but any substance. Okay, the substance required for living organisms. Yes, many substances are there which need by living organisms. Okay, the substance that are required for the living organisms for what? For growth. For growth, we need some substances. They are what? Primary metabolites. What are they? What are we need for the growth? That is what? Growth, proteins, carbohydrates, fat. Example, we need proteins, carbohydrates, fats, amino acids. All we are taking in the form of diet, right? Amino acids, nucleic acids, etc. These are found in all organs. Substances are not required. Secondary substances which are not required for living organisms. Required for living organism. Substances some are required for living organism are called primary. Secondary metabolites are nothing but what? Secondary metabolites, the substances which are not required for the living organisms. Okay, however, many of them are useful for human welfare. Some are there. Rubber, drugs, example, rubber, drugs, pigments, spices. Spices are some of the examples. Okay. Next class we have one tabular column that we will see in the next class. Okay. So any doubts what we have studied? What we have studied, that is biomolecules, how to analyze chemical. Now we want to know in tissue what all chemicals are there. First it should be treated with the trichloroacetic acid. We will get two fractions. One is filtrate, another one is retentate. One is soluble, another is acid insoluble. Okay, we have analyzed. And primary and secondary metabolism is taking place. Any living organism, they undergo growth. So for growth, we need some food substances. We need protein, carbohydrate, fat, amino acid, nucleic acid for the growth. But secondary metabolites, which substances which are not required for the living organisms, drug, pigments, spices, or it may be the rubbers. Okay. Next class, we will list off some chemicals. Okay, after that the elements, percentage, how much percentage it should be present in the living cell, all that list we will do in the next. So, micromolecules, macromolecules differences you are going to see there, what we have studied. Micro and macromolecules. Introduction. Before going to analysis, you should know micromolecules, macromolecules. Micromolecules are forming by condensation. They are going to convert into macromolecules based on the size of biomolecules. Two types based on the size, particularly micro. These are micro means small, macro means large. Okay. So low molecular weight, high molecular, here high solubility, then low solubility. These are simple structure, they are very complex structure. Okay. These are found in different way. These are found by the polymerization of micromolecules or condensation of micromolecules. Okay, both they can exist, they may be organic and inorganic. These are particularly organic. Okay, they cannot pass the cell membrane, which can easily pass the cell membrane, water, minerals, amino acid, sugar, phosphate, etc. These are proteins, nucleic acid, polysaccharides. Okay, these are all types. So what we have completed, biomolecules introduction, how to analyze chemical composition, and primary and secondary metabolites we have completed. Then next class there will be change from tomorrow onwards. The class will begin in the Zoom. Okay, everyone, you should attend the class compulsorily, and that will be forwarded in your WhatsApp group. Okay. So if you have any doubts, you can ask some question. Biomolecules chapter is very important. You can ask the questions in your group.